Hi everybody. Today we discuss an important topic which is called as phospholipids. Phospholipids. It is important because it could be a short note for UG as well as PG students, and it comes in MCQs also. Okay. Now what is phospholipids? If we can understand that it has a phosphorus group and it is containing lipid, so we can say that it is a compound lipid. It is a compound lipid which contains phosphoric acid. And this phosphoric acid provides the phosphorus that is required in the formation of phospholipids. Okay, so in simple terms, it is compound lipids and it contains phosphoric acid which provides the phosphorus group, and this is how the phospholipid name justifies the presence of phosphorus. Now it is not that simple also. It just does not contain only phosphorus plus the lipid group. It is divided into two categories, and these two categories have difference in the backbone of their structure. This means they have a different backbone, means they have a different moiety altogether over here. So this is the difference. The backbone is different for these two structures. So depending upon the backbone, they are given the name as glycerol. This is called as glycerophospholipid, or phosphoglyceride. This means glycerophospholipid or phosphoglyceride. This means the backbone over here in this first group is glycerol. Okay, and the second one is called as sphingospholipids. So this means the backbone of the structure contains sphingosine. Okay, sphingosine. So the presence of the alcohol is different. One which contains glycerol is called as glycerophospholipid or phosphoglyceride. The one which contains sphingosine is called as sphingophospholipid. Now what is the difference? See, if you see, the glycerol I have drawn a straight line and sphingosine I have drawn a reverse L. So the structure is different. Okay, sphingosine the structure is different, the glycerol the structure is different. So glycerol gives me binding to extra 3 moieties. So in the first carbon atom, it will bind to a fatty acid. In second carbon atom, it will bind to a fatty acid, and in third carbon atom, it will bind to phosphoric acid, the phosphorus, and to a base, which is generally a nitrogen containing base. So it is a nitrogenous base. Why I am telling is on this in such a simplified way, because see, remembering the structure is not that important, because we are not genius enough material to remember structures also, to which carbon atom which is attached and how it is formed. So basically, if you know what is present, how in a very simplified diagram, it makes things easier. And this structure also you can draw in the exam. Nobody expects you to know which carbon atom is where. We deal with biochemistry and we do not deal with chemistry. If you can remember the structure, well enough. But on my personal opinion, even I don't remember the structure. So I cannot comment that you should also remember the structure, because I don't remember the structure. Now coming to sphingosine. Sphingosine is different. Why? Because it is a amino alcohol. So the first difference is it is an amino alcohol. Now what will happen? This outlet is there in the second part. The first is sphingosine, it has a reverse S structure. So this second part is attached to a fatty acid, and the third part is again attached to a phosphoric acid from the phosphorus from the phosphoric acid and a base. So can you now see the difference in the structure? And the difference one more is that the amide linkage is present when it is attached to the fatty acid. So there is an amide linkage, then it is attached to the fatty acid. Okay, so sphingosine and glycerol phospholipid and sphingophospholipid differ in these base. One more thing that we're supposed to remember the structure is that the fatty acid, the first one is always a saturated fatty acid and the second one is always an unsaturated fatty acid. Okay, unsaturated fatty acid. Now, if we summarize phospholipids, there are two types: glycerophospholipid or phosphoglyceride and sphingosphospholipids. So glycerophospholipids contain the glycerol as the backbone and they are attached to two fatty acids. That first carbon is saturated, second carbon is unsaturated, third there is a phosphorus group which has attached to a nitrogenous base. Second sphingophospholipid is a sphingosine, that is an amino alcohol. It is attached to fatty acid through its amide linkage and there is a phosphorus and there is a base. Now one more important point to remember in this structure is that this sphingosine, that is sphingosine plus fatty acid, this whole structure, there is again a name given to it and it is called as a ceramide. Okay, so ceramide is sphingosine plus fatty acid is a ceramide, or we can say a sphingophospholipid minus the phosphorus minus the base is a ceramide. Okay, and another thing that I have been telling, there is no glycerol. Okay, so there is no glycerol altogether in sphingospholipids. If this is clear, then now we go further in what are the different types of glycerophospholipids and sphingospholipids. Now coming to first important thing is, why do you want to even learn biochemistry when it is thought that it is not that important a subject? So first we will see why are we studying phospholipids. Phospholipids would be important somewhere, that is why we are learning it. Otherwise, who cares to learn things which are not important? So phospholipid, what is the clinical application of phospholipid? First, it is present in the membrane bilayer. It is present in the membrane bilayer. No membrane bilayer is complete without phospholipids. It is present in membrane bilayer. And then if you have listened to the lipoprotein lecture or lipoprotein story, so it is present in lipoproteins. It helps in fat absorption. And it is an important some are important lipotropic factors. Lipotropic factors means that help in the prevention of fatty liver. Fifth, another important point is that some of it is a surface, it reduces the surface tension, and if it is not present, it may lead to respiratory distress syndrome. And when I say it is important in lipoproteins, fat absorption, the other important point that we are supposed to remember is that it is also a part of reverse cholesterol transport. So wherever there is a biochemistry, in all cycles, in all metabolisms, there is biochemistry, and everywhere there is phospholipid because it is present in membrane bilayer. It is present in lipoproteins, in fat absorption and reverse cholesterol transport. It is important as a lipotropic factor. It decreases the surface tension, and if I can also say that it is also important in nerve transmission. Some of them are also important in nerve transmission. So phospholipid is not supposed to be ignored, and it is not ignored by the examiner also, and hence it is a very important short question which is always asked. Okay. Now since we have seen why it is important, now we will see all molecules where it plays an important role. The first one is a very simple phospholipid, that is phosphatidic acid. Okay, so this is phosphatidic acid, so it is a glycerol molecule, that is a backbone. It contains a fatty acid, one more fatty acid, and it contains only phosphatidic acid. Okay, so it is a simple form. It is basically intermediate in the metabolism, not present everywhere. Okay, so we have discussed phosphatidic acid. Second is phosphatidylcholine. Now phosphatidylcholine, what will happen? Here there will be a base. So what is the nitrogenous base over here? Choline. So this is the nitrogenous base over here, and this would be phosphatidylcholine. Phosphatidylcholine is also called as lecithin. So it is also known as lecithin, and phosphatidylcholine also known as lecithin, containing a nitrogenous base which is called as choline, and this one is important in nerve transmission. Why? Because choline helps in the formation of acetylcholine. Okay, helps in the formation of acetylcholine. It is an important part of biological cell membrane, and is involved in nerve transmission, and it is, if you clearly see the structure, it has methyl groups. So if required, it can give the methyl group, but in biochemistry, as I say, there is only Mr. Sam who gives us the methyl group, S-adenosine methionine. But if required, it contains labile methyl groups. Choline contains labile methyl groups, and it can give if required. Now coming to the third, it is dipalmitol. No, it is the part of lecithin only. Dipalmitoyl lecithin. So what is the function of dipalmitol lecithin? It is present in the lungs, and this is the one which acts as a surfactant, and if this is not there, it causes respiratory distress syndrome in newborns. Okay. Other important part of choline or phosphatidylcholine or lecithin is to remember is lysolecithin. Lysolecithin. If you remember, lysolecithin we have discussed in the metabolism of high density lipoprotein, where lysolecithin was formed, and this is a marker of atherosclerosis. How? Because it gets deposited in oxidized lipoproteins. It gets deposited in oxidized lipoproteins. Okay. So till now we have discussed two phospholipids. Phosphatidic acid is intermediate in metabolisms. It is very simple. It just contains a phosphatidic acid group. Phosphatidylcholine, also known as lecithin, important in nerve transmission membrane, biological membranes, has methyl groups. The second part, that is the subgroup of phosphatidylcholine, dipalmitol lecithin, it is present as surfactant. Lysolecithin is present in oxidized lipoproteins and is marker of atherosclerosis. So 2 we have discussed, 5 more to go. As I always say, remember the number. When you remember the number, you all obviously remember how much you have to write. Okay. So 2 we have done. Now coming to the third. Third is called as cephalin. Now cephalin will contain phosphatidyl ethanolamine. Ethanolamine. If you have difficulty in remembering what is cephalin, so E is the second word, and our second alphabet, and our second word starts with E. So instead of choline over here, you have ethanolamine. Okay, you have ethanolamine. Now what is the importance of ethanolamine? It is helpful in blood clotting mechanism. Okay. Now coming to the number 4. Number 4 is called as phosphatidyl ionositol. Ionositol. It has containing the stereoisomer myoinositol. Even I get confused when spelling sometimes. Myoinositol. So over here, instead of ethanolamine, now you have myoinositol. Myoinositol. Now this is very important, as we see, because it acts as a second messenger. How it acts as a second messenger? Because it will break down into inositol phosphate and triacyl glycerol. So it is important in giving the messaging service of hormones. The fifth one is phosphatidylserine, phosphatidylthreonine. Just the substitution over here will be serine, here the threonine. The sixth one is plasmalogen. Plasmalogen. Now what is the difference between other phospholipids and plasmalogen is that it contains an ether linkage. it contains an ether linkage. So we have to remember this, that it contains an ether linkage at the C1 carbon atom. So all the rest remaining the same. Here now it would be a ether linkage if there is a plasmalogen. Okay. So if it is a plasmalogen, there would be an ether linkage. Now it could be inositol. So it would be called as phosphatidyl inositol. We always call it as phosphatidyl. Okay. But when it is a plasmalogen, it is an ether linkage. It is called as phosphatidyl. The nitrogenous base could be anything. It could be choline, phosphatidyl choline. It could be phosphatidyl ethanolamine, phosphatidyl inositol. Okay. Now coming to the last one. That is a name which is called as cardiolipin. And we always think that it is present in cardiac muscles. But it was isolated first from cardiac muscle. But it is very important part of mitochondrial membrane. So what we have to remember is that it is an important part of mitochondrial membrane. Okay, a mitochondrial membrane. So there are two moieties of phospholipid, two moieties of phosphoric acid, and that means there are two molecules or two moieties of phosphoric acid. That is the difference. Two moieties of phosphoric acid. Okay. So we have discussed with phospholipids. Only the nitrogenous base keeps on changing. When you are having a plasmalogen, there is an ether linkage. So this is very clear now. So if we have to summarize, what are the phospholipids present? Just remember the number 7. So phospholipids, if in short we say, it will be glycerophospholipids. Phospholipids, it would be glycerophospholipids, it would be sphingospholipids. In glycerophospholipids, you have phosphatidic acid, second you have phosphatidyl choline, third you have phosphatidyl ethanolamine, fourth you have phosphatidyl inositol, fifth you have phosphatidyl serine, phosphatidyl threonine, sixth you have a plasmalogen, and seventh you have a cardiolipin. So basically it is easy to remember because you have done with you have not done, if some of you have studied pharmacology, so like ACI inhibitors we say, so phosphatidyl acid, phosphatidyl choline, phosphatidyl ethanolamine, phosphatidyl inositol. So ACI inhibitors, if you could just remember ACI inhibitors, so it could be serine and threonine, and plasmalogen, where everything is converted into ether linkage. The above will be converted into ether linkage, and the most important of all, that is the cardiolipin. Okay, that is the cardiolipin. So this is the way. If you want, you can remember. Otherwise, it is a bit difficult. You can forget on one or two. The most important part is that it is an important part of biological membrane. Once you remember that it is an important part of biological membrane, other things become easy, lipid transportation and all that. Now coming to sphingospholipids. Sphingospholipids as such is important in formation of whatever it reminds you, sphingomyelin. It reminds you of sphingomyelin. So it is important in the formation of myelin, which is present in brain and nervous tissue. Okay, the ceramide, as we had discussed, again I will draw it. There was a sphingosine. There was a fatty acid which was at amide linkage, and then there was phosphatidic acid, and then there was a base. So this all was called as ceramide. So this ceramide is also important because it is a second messenger. Like phosphatidyl inositol, we had a second messenger. Ceramide is also a second messenger. But what is important is, where is it a second messenger? Remember it very carefully. Second messenger in apoptosis. So cell cycle, cell differentiation, okay, And sometimes there could be a 30 carbon fatty acid which is attached over here, and it is an important in giving skin permeability. So that is another point altogether. Ceramide is important in apoptosis, in cell cycle and cell differentiation. For basically all this we are supposed to remember in phospholipids. It is a simple topic if you just remember the structure, and you just need to keep on changing the base over here. That's all for phospholipids. Have a good day. Thank you, and please save water. People are in need of it. I am in need of it. Thank you.